The next topic we're going to talk about is the structure of DNA. DNA, also known as deoxyribonucleic acid, is a double strand of nucleotides in a twisted ladder shape called a double helix. So you will notice that it has two sides, and that if we were to untwist it, we would be able to see kind of like ladder rungs, okay? It's really important that you know that it's two strands, and it's also really important that you know what DNA stands for, deoxyribonucleic acid. The nucleotides of DNA are made up of the sugar deoxyribose, a phosphate, and a base. We know that all nucleotides are made up of a sugar, phosphate, and base, but this particular nucleotide has the sugar deoxyribose. The sides or backbone of the ladder are alternating sugars and phosphates and are held together by strong covalent bonds. Why do you think you might want the backbone held together by strong bonds? Think about your backbone. For the most part, your backbone is the support for the rest of your structure, and that's the same for DNA. So if we take a look, we have a dot, and then this light blue figure, dot, light blue figure, dot. The dot is the phosphate, and our light blue figure is the sugar. The steps of the ladder, or the rungs, are formed by the nitrogenous bases. These are held together by weak bonds called hydrogen bonds. Why might we want the steps of the ladder held together by weak bonds? Well, think about what we needed to do before mitosis began. We had to go through interphase. And one of the parts of interphase was S phase, where we had to make a copy of our DNA. In order to copy the DNA, we need to be able to pull it apart. So in order to pull it apart, we really want some weak bonds there so that it doesn't take too much energy. Think back. When have you encountered weak hydrogen bonds before? If you were thinking when we talked about water, you'd be correct. Hydrogen bonds were the type of bonds that allowed surface tension to be created on water. The nitrogenous bases in DNA are adenine, A, thymine, T, cytosine, C, and guanine, G. It's important that you know which four bases are found in DNA. The purines are the larger bases, which are made up of two rings. These are going to be adenine and guanine. The pyrimidines are the smaller bases, which are made up of one ring, thymine and cytosine. So if we take a look, we have two rings in purple. This two-ring structure would be either adenine or guanine. The single ring structure that's in blue on the right-hand side would be either thymine or cytosine. So in order to keep the width of the DNA molecule the same, we always have a larger base paired with a smaller base. This is called complementary base pairing. So A will always pair with T, and G will always pair with C. When A pairs with T, you are going to have two hydrogen bonds, as you can see in the figure on the right. When G pairs with C, you are going to have three hydrogen bonds, as you can see in the purple and blue figure on the right. So it's important to know how many bonds are formed between each of the bases and what the bond type is. So here's a question. If 30% of the bases are adenine, how many or how much is cytosine? Well, if I have 30% adenine, there must also be 30% of thymine or T. This gives me a total of 60% of my DNA. The other 40% is made up of C's and G's. So if I take my 40% and divide it by 2, that means 20% is cytosine. This is Chargaff's rule. So Chargaff stated that A pairs with T and G pairs with C. So it's through his rule that we were able to come up with the amount of each type of base that's found in DNA. Next, we're going to talk about the structure of RNA. RNA stands for ribonucleic acid. It's important that you know what RNA stands for. RNA is a single strand of nucleotides as opposed to DNA, which was a double strand. The nucleotides of RNA are made up of the sugar ribose, a phosphate, and a base. So the sugar in RNA is different than the sugar in DNA. Remember, DNA was deoxyribose and RNA is ribose. The nitrogenous bases in RNA are pretty much the same as DNA, except for uracil, U, is going to replace thymine. So we are still going to have adenine, cytosine, and guanine. In this case, A is going to pair with U and C is going to pair with G. It's important to note that U's will only be present in RNA. So if we take a look at the differences and similarities between DNA and RNA, first we're going to look at the structure, the function. The function of DNA is to be the instructions for everything that the body needs, and it's kept in the nucleus. The function of RNA is to bring the code out to the cytoplasm and make proteins from it. So, DNA is essentially my master plan, and RNA is the blueprint. So if we were talking about architects, for example, the master plan that stays at their office would be DNA, and the blueprint that they bring out to the work site that might get rained on or might get some mud on it or it could possibly get destroyed is my RNA. My DNA has 2 strands and RNA has 1. The sugar, which I think you might have to write into your notes because it got cut off by the hole punch, is deoxyribose in DNA and ribose in RNA. And the bases are pretty much the same except for I'm going to have T, thymine, in DNA and U, uracil, in RNA. There are 3 types of RNA. The first one we're going to talk about is messenger RNA. Messenger RNA, or mRNA, is responsible for copying the DNA's genetic code. It then carries the genetic information out of the nucleus to the ribosomes, meaning that I will find mRNA in my nucleus, at a ribosome, or somewhere in my cytoplasm. mRNA carries the information for a specific protein, and the letters or bases in mRNA make up the words called codons. Each codon is 3 bases long and codes for 1 amino acid. So every 3 mRNA bases codes for 1 amino acid. The second type we're going to talk about is ribosomal RNA, or rRNA. rRNA is probably the easiest of the 3, as its only job is to make up the ribosome. The last one we're going to talk about is tRNA, or transfer RNA. Transfer RNA is going to come and match up or complement my mRNA. In doing so, it's going to drop off an amino acid at the ribosome where the proteins are going to be made. If I look at my tRNA, the shape is a little bit like a clover leaf. The top part has the attachment site for the amino acid, and the bottom part has an anticodon, or the part that's going to complement my mRNA.